Hello class. So in this video, we're right in the middle of chapter 16, talking about our non-specific defenses. And we're going to talk about phagocytosis, or the way that our white blood cells just chomp up any pathogens. And we're going to talk about inflammation, or what happens if you get a small cut or something that could be infected. So phagocytosis is Greek for cell eat. So phago means eat, cyte means cell. So it's cell eating. This could include white blood cells that are going to ingest microbes that are going to eat any pathogens or eat their particles. Although phagocytosis is important also for removing dead cells and removing denatured proteins. So it gets rid of other stuff that we don't need cluttering up our body. In the process of phagocytosis, first the cell will extend a pseudopod, and that pseudopod is able to adhere. First it starts with a toll-like receptor binding to a PAMP. We talked about that. Your cell will extend a pseudopod that will kind of wrap around the microbe and bring this microbe in, just kind of hug it on in. When that microbe is inside of a cell, this vesicle is called a phagosome. That is the phagocytic vesicle, where that microbe has been phagocytosed, but it's still sitting there like it was before. That phagosome can fuse with a lysosome. If you remember, lysosomes have all kinds of fabulous digestive enzymes. So it has all those enzymes that can break stuff apart. When the lysosome and the phagosome fuse with each other, they form what's called a phagolysosome. And all these digestive enzymes are going to start breaking down that microbe. So then your microbe is going to be broken down as far as it can be broken down. Any indigestible little bits are just going to be kicked out to be cleared out of your body. And that microbe has now been destroyed. If you remember from chapter 15, we did discuss a few ways that microbes can evade phagocytosis. So M proteins and capsules can help inhibit the adherence, so inhibit the ability of those pseudopods to adhere to a bacteria. Leukocidins can kill phagocytes. So as a phagocytic cell comes up and starts a process, the bacteria can kill the phagocytic cell. Membrane attack complex can lyse phagocytes. Some bacteria can escape the phagosome. So they're not able to be trapped inside of that phagosome. They just get right back out. Some can prevent the phagosome from fusing with the lysosome. And so these bacteria, HIV and mycobacteria, or the virus HIV and the bacteria and mycobacteria, are able to just sit there and survive inside of that phagosome because it's not been fused with the lysosome. And then Coxiella burnetii is able to survive in the phagolysosome. So those lysosome enzymes are not able to break down that bacteria. Oh, sorry, I should take that first question out. We took out fixed and wandering macrophages. But tell me, what is the role of TLRs in phagocytosis? You should be able to tell me what is a phagosome, what is a phagolysosome, what is a pseudopod. Be able to just kind of diagram out that whole process of what's happening through the process of phagocytosis. Then in inflammation is a local response. So this is not an infection throughout your body. This is you stabbed yourself with a knife, you have a small infection on your thumb, a local defensive response to an injury or to an infection. It functions first to destroy the microbe and any byproducts of that microbe. It also functions to limit the effects on the body. So it'll stop the bacteria from spreading through your body and causing damage in other parts of your body. And it'll repair the damaged tissue. So, that thumb that you just stabbed with the knife is going to be repaired so that you don't have that wound forever. Inflammation is going to rely on histamine. Remember talking about histamine released from our basophils earlier? So histamine does two important things in the process of inflammation. One is cause vasodilation. Vasodilation is the increasing the dilation of blood vessels, which means that more blood can come to that area. More of your immune system is going to be able to come to that area of infection. It also increases the vessel permeability. So once those cells are in the infected area, they can get out of the vessel, so they can leave the vessel and get into the infected tissue and mount an immune response. There are 4 cardinal signs and symptoms of inflammation, and I imagine you could probably already tell me these, but we're going to go through it. Redness, heat, swelling, and pain. Redness, or rubor sometimes, is due to an increased blood flow. So as more blood is flowing after that vasodilation caused by histamine, that more blood is red, so it's going to turn your area slightly red. Heat, or calor, related to white blood cells acting, so that physical action is going to produce heat. And also having more blood that is warm is going to produce heat. Swelling or tumor, because you have more cells and fluid in that area. They take up space, so they swell. Edema refers to an accumulation of fluid. Pain or dolor is a result of your nerve endings being stimulated because prostaglandin's been released. We didn't talk about that a lot, but it comes with the process. Sometimes a 5th symptom is thrown in, which is a loss of function. So, if you get cut on your knuckle, you might not be able to bend your knuckle. You might just have a temporary loss of function in whatever body part has been injured. Okay, so during the process of inflammation, so here is the initial localized wound. You're cooking, chopping up chicken, and accidentally stab yourself. So there's lots of bacteria from the chicken on that knife. When that stab happens, chemicals are going to be released. Histamine, prostaglandin, cytokines. Those are probably the important ones we've talked about. Histamine, prostaglandins, and cytokines are released by the damaged cells. You'll start forming a blood clot. That blood clot is important because it is going to trap these microbes within the clot so they're not spreading. Then your blood vessels are going to be vasodilated and have that blood flow coming through. And they're also going to be more permeable to allowing your white blood cells to come through. 2 important terms here. One is margination. So I think of margarine sticks to toast. Margination is your phagocytes stick to the endothelium. Your white blood cells stick to the lining of your vessels. So they're going to slow down instead of spreading, just flowing on through your blood. As they start to marginate or start to stick, those white blood cells are going to start to adhere and slow down. They do what's called leukocyte rolling, where they're getting ready to come out, and then they perform diapedesis, which is when those white blood cells actually squeeze between those endothelial cells. So they're coming out of your blood vessel into the infected tissue. Then when they're in the infected tissue, your leukocytes are going to start chomping up, doing phagocytosis and eating all of those infectious particles. So you can describe to me, what is the purpose of inflammation? What causes the redness, swelling, and pain associated with inflammation? And what is margination? Be able to define margination, be able to describe diapedesis. All right, we still got a little bit more of this chapter to cover. We need to talk about fever and other ways that our body is going to protect us from infection. See you back in a little bit. Bye.